Hey, chemistry team, ready for video number 6 in our journey through acid, base, titrations, buffers, and common ion effect and all that fun stuff. So we're focusing on titrations right now, which is just application of those earlier concepts. Before I draw up a titration curve, which is probably new to most of you, like I said, you've done titrations, calculated unknown concentrations, but I don't think you've ever done a titration curve and gone, wow, I wonder what the pH is at any point along this titration process. Here's our focus. We're going to make graphs. Yay, graphs, right? Get your Microsoft Excel ready to go. We're going to plot the titrant volume, right? Remember, if you're in lab, you usually control one of two parameters. So like if you're doing density and temperature, you control the temperature, not the density. So the one that you control goes in the x-axis. That would be temperature, for example. And the density depends on it, so the density goes in the y-axis. So the y-axis is called the dependent variable. It depends on the other variable called the independent one. So if you change the independent variable, temperature, density changes in response. That's the dependent variable. That's always the y-axis. So if you're ever not sure what to plot where, the one you control goes in the x-axis. So in this case, we don't control the pH, we control the volume of titrant we squirt out of the burette. So we take the titrant volume, that's the independent variable, in milliliters, that goes on the x-axis. As the pH changes, right, as we add more base, for example, the pH will go up. That's going to go on the y-axis. That would be the dependent variable. And the pH, of course, has no units. We could do the pOH, but that's kind of weird thinking. It's all like backwards in our brain. So I very, very, very rarely ever see a pOH versus titrant volume. It's almost always pH. But we could if you're bored and got nothing to do. Our focus, though, will be at the very, very, very small amount of volume. Remember when you're in lab and you're adding a drop here or a half a drop, trying to get that faintest endpoint color? The pH changes dramatically. Even one drop of a strong base near the equivalence point, boom, that pH goes crazy and changes more than you could possibly conceptualize right now. And we'll see that when we do the graphs and do all these plots and stuff. So our focus is right around that equivalence point to see how that pH just changes dramatically. All right? And of course, if you're adding a base to an acid, you're going from a lower pH to a higher pH. If you're adding an acid to a base, you're starting at a high pH and going to a low pH. It depends what you're starting with. But most people, you're adding a base to an acid. That's typically how it's going to happen. So you start at the pH of the acid. pH goes up, 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 hits the equivalence point, and then keeps going up if you go past. Although in lab, we don't go past the endpoint, but theoretically we could. I'm going to teach you how to calculate the pH at any single point along that titration curve. I know you're so excited to do that. I actually took my Microsoft Excel and I typed in the equations, put those functions in there, and I could just type in the concentration of my base and acid and the volumes, and it just made the plots myself, made these beautiful titration curves. All right, so let's look at we're going to look at the details for the different types, weak acid, weak base, strong acid, weak base, strong acid, strong base. They all have different types of titration curves, but they follow a similar pattern. So let me show you a generic titration curve and where the different regions are and the types of calculations we're going to need to review, if you're a little rusty on them, to be able to calculate the pH at those points. So what exists in those particular areas. So let's draw a titration curve next. Here's what you're going to see generically, right? So each individual titration curve will be similar in form to this, but the specific locations, the specific pHs at certain volumes will change if you're doing a strong acid with strong base or a weak acid with a strong base, and vice versa. Again, if you're starting with a base, you start up at a high pH and come down. Remember, we're plotting our titrant volume on the bottom. That's on the x-axis. That's typically going to be your base, typically. And then the pH at the top, on the left-hand side, on the y-axis, going from like a 1 to a 14 is typical, or 0 to 14. Very rarely do you get below 1. You got to have some majorly strong acid for that. So typically 1 to 14 or 2 to 12, something like that's going to be perfectly fine for your y-axis. Obviously, we're going to have 0 to start with. And if you're using a burette in lab, you ain't going past 50. If you do, you went below the bottom line and you're toast. You can't do that. But theoretically, we could have a huge burette. Well, do you see the 4 different regions here or areas? Now in blue, I put specific points, really critical points. And then in the light brown, I put regions. So if we are starting with just an acid and we haven't added any base from a burette, it's just the acid sitting in the Erlenmeyer flask. We're going to be at this blue dot right here. We have added 0 titrant, so it's going to be at some initial pH. And I'm going to talk about on the next board how to calculate that generically. So we'll have some initial pH, either of a weak acid or a weak base, or technically we could have like a strong acid, strong acid or weak acid. And if we started with a base, we could have a strong base or a weak base, which would start up here, right above 7. We titrate, we titrate, we titrate. We're adding base to, in this scenario, base to the acid, but we still have more acid than base. Anything before the equivalence point, you have more acid than base. So we call this the pre-equivalence or pre-equivalence point region. Any amount of base volume added before we hit the equivalence point, right? Where we still have a little more acid than base. There's going to be specific types of calculations in those regions. Then, of course, we've got the equivalence point. That is when the acid and base have exactly neutralized each other. The number of equivalents equals the number of equivalents of base and acid, right? So the same number of equivalents. That's going to have a very specific pH depending on what the acid and bases are. And I'm going to show you. Those are the toughest calculations right there, trying to figure that out. So very heavily dependent on what those acids and bases are. Now, anything past the equivalence point, we have more base than acid. Right? So obviously the pH will continue to rise. So we call that the post-equivalence region. Fairly simple calculations in that region because all the acid's gone. So no matter what kind of titration you have, you just have excess base. So it almost becomes like a limiting reactant problem. You go, well, how much base did I add? Right? How much acid did I start with? I neutralized all the acid. So what's the excess base that I have? And you can just calculate the pOH and the pH from that. So the post-equivalence region to me is the easiest region. You've learned everything you need to do the initial pH. You've learned everything you need for the post-equivalence region, the easier ones. Equivalence point's a little tricky, but you know how to do it. I'll have to review some of the concepts. The pre-equivalence region really depends if it's a strong acid or a weak acid, strong base, weak base. That'll tell you what you formed in those regions. Really, your thinking is, what exists here? Not just the acid, right? What exists here? Well, all the base is gone, so just the acid, and then any products from that particular neutralization. What exists here? Well, whatever the products are. You have no acid or base left, so what are the products of that neutralization? What exists here? Only the base, right? Because all the acid's gone. So if you can really hone in on what are the predominant species existing at that particular point along that titration curve, we can then calculate how they interact with water and what the pH would be. There's going to be a lot of review on this one. A lot of acid-base reviews like ions, acidic or basic in solution, those kinds of things. So let me briefly talk about each region, and then we're done with titration curves, and we'll do indicators in another region because we want to pick it. Now notice how it just skyrockets. Right, so it's kind of like la-di-da, not much happening. You're titrating away, not much happening. And you've probably seen this in lab. Nothing, nothing, nothing. You add one drop, boom, pink. You're like, wow, I only added like a drop. And that one drop skyrockets that pH, right? The acidity changes thousands or 10,000-fold, maybe even 100,000-fold with like one stupid drop. So you'll take this tiny little bit of volume, a drop or two, and the pH just goes boom. And then it kind of tails off after the point. So you get kind of this, if you've ever, looked into S-curve economics, this kind of looks like an S-curve in S-curve economics. Cool thing to research. If you're bored and have nothing to do, you can Google S-curve economics or buy yourself an economics textbook and review S-curve economics. Really neat. See how products and services launch into society with pioneers and, laggers. Anyway, why am I talking about economics? But you kind of see it looks like a letter S. We are really focused on the pH change here. So if we want to pick an indicator that changes color, ideally right at the equivalence point pH, but see how steep that slope is? We really could pick an indicator that changes color anywhere in that whole region right there. Right? It's just boom, it's a vertical line. Good luck scaling that one. So as long as the indicator changes color, we'll see within a certain number of pH units around that equivalence point, we're good. So there might actually be 3 or 4 different indicators that work perfectly well. We'll just pick one that, in the lab, we look at a monetary cost, disposal, environmental disposal issues, colors. Like I always like going from a colorless to a color versus going from like yellow to green. Colorless to green is easier than yellow to green. Well, anyway, let's look at each region and the types of calculations and how you can review the concepts if you're a little rusty on those. That would be good to go. Here's the 4 regions we looked at on the titration curve, and just a way to think about it. And again, we're going to do videos for each case: strong acid plus strong base titration, weak acid plus strong base titration, yada, yada, yada. And we'll look at these in specific detail and do an example. That's going to take a video for each one. Initial pH, nothing new. You're just looking at, if you're starting with an acid, is that acid strong or is it weak? If it's a strong acid, right, 100% ionization, so the concentration of the hydronium ion is the concentration of the original acid. Take the negative log of that, boom. If it's a weak acid, equilibrium, set up your ice table. So if you're a little rusty on that, go back to weak acids and weak bases and the equilibrium that they are set up, and how to use the Ka and Kb values, right? Depending if you have a strong, starting with a weak base or a weak acid. But do the equilibrium if it's weak to figure out the, calculate X from the ice table, and X will either be OH minus for a weak base or H3O plus for a weak acid, and then take the negative log of that to get the POH or pH. If it's a POH, subtract it from 14 to get the pH. If you don't have the faintest idea of what I'm saying right now, go back and review those videos. You're going to need it. You're going to do a lot of these, my friends. And that's just the first data point at 0 volume, 0 titrant volume. Now, anything between 0 and so you added even the faintest drop past the initial amount up to even the faintest portion of the drop before the equivalence point. That's the pre-equivalence region. The types of calculations, it's always going to be based on neutralization. I have this amount of acid in my flask. I've added this volume and concentration of base. All the base goes bye-bye. How much acid is left? Or if I'm starting with a base and adding acid, all the acid goes bye-bye. How much base do I have left over? The titrant from the burette's gone because you have less of it. Okay, so you got to do the stoichiometry neutralization and go, how much of the starting species, whether it's an acid or base, is left in my flask after I added that specific amount in concentration of titrant volume. Once you've done that, look at what exists. So if I'm titrating a strong acid or strong base, all I'm going to have left is that strong species. Well, then you can just take the negative log of that concentration, you're good to go. But if I'm titrating a weak acid or a weak base, oh no. If I have a weak acid and I add a smaller amount of a strong base, that creates a buffer. If I have a weak base and I add a smaller amount of strong acid, that creates a buffer. So you're going to have to do the Henderson-Hasselbalch equation. Ha 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 ha. So a lot of people call that pre-equivalence region a buffer region if you're starting with, if one of your 2 species is weak. That's a little bit trickier. Lots of stoichiometry neutralization. So I would review buffers, which is one of the earlier videos from this particular chapter. So look up, review the common ion effect video and the buffer video. Going to be very, very helpful for the pre-equivalence region only. At the equivalence point, remember, all the acid and base are gone. Doesn't matter if they're strong or weak. They have completely neutralized each other. Bye-bye. So all you have left, acid plus base, gives you salt plus water. Water's just neutral, so we don't have to worry about the water. That's pH 7. But the salt is the cation and an anion. So you need to go back and review the video on acidic and basic behavior of ions that are in solution. So the salts hydrolyze, break apart in the cation, anion. They solvate, right? Does that anion or does that cation have weak acid or weak base behavior? Oh no! Go review those videos. You're going to need to. And again, I consider the equivalence point the most challenging one to calculate because it takes a lot. You're going to see like almost an entire page on your final exam just for that equivalence point. Post-equivalence region, now remember, if you started with acid, you've neutralized it with the base, all the acid's gone now, and now if we keep adding base, all we have left is the base solution. So if that's a strong base, that you just figure out using neutralization stoichiometry, right? How much acid did I start with? How much base did I add, right? How much base was used up to neutralize the acid? How much base is left over? Right? Calculate your OH minus concentration. Take the negative log of that. That's POH. Subtract that from 14. Wada bing, wada boom, baby. I got my pH. And vice versa, if I started with a base and added acid to it, that base is gone at the equivalence point. I'll have left over acid. Usually it's strong, and then you just do the negative log of the pH. But almost always we start with the acid. But like you see, it depends on what you're adding to what. Is it strong or weak? Are you starting with acid or are you starting with base? Not even looking at polyprotic species, and we're not going to do weak acid, weak base. Too challenging. So again, I think the post-equivalence region is the easiest. You just do the neutralization stoichiometry, how much of the titrant's left over, and since it's almost always a strong species, it's just a negative log of what the remaining concentration of that is. But not too bad, right? So we'll do indicators. How do you know which indicator is best for a particular type of titration? So we'll cover that, and then we'll get into specific ones. We'll do strong acid plus strong base titrations in a video. We'll do weak acid plus strong base titration video. We'll do a weak base strong acid titration video, and maybe touch a little bit on polyprotic weak acids with strong bases, and we're done like forever with this class. Yay!